Hello and welcome to my RCAW talk. My name is Sarah Goodnight. I'm a PhD student here at ECU, and today I'll be talking about my dissertation research titled, "Do Cannibals Make Good Hosts? Cannibalism Drives Parasite Abundance Patterns in Larval Dragonfly Communities." So to get right into it, parasites are incredibly diverse and exist across a diversity of taxa. So we have insect parasites. We, of course, have a wide variety of worms. And of course, we have our lovely tongue-eating isopod, a very charismatic parasite of fish. Now, the thing about parasites is that they are dependent on their hosts. So host interactions are going to be the main drivers of parasite population structure and transmission. And we have direct interactions like predation and cannibalism, which transmit parasites from prey to predator. So those are very important, of course, but we also have more indirect processes going on. So things like host presence and host population density are going to affect things like transmission probability and infection risk in a community. So I want to get right into cannibalism, which is going to be a big focus of my talk today, which occurs when individuals of the same species consume each other as food. Now, cannibalism can actually be a very important structuring force in many predator communities, and generally it is actually considered to be detrimental to parasites, especially in a single host kind of simple parasite context, because cannibalism eliminates viable hosts without actually increasing the number of infected hosts. So one host might eat another one and become infected, but that didn't actually increase the number of infected hosts in the population. Now, this idea of cannibalism as detrimental to parasite transmission has really only been evaluated in a single host, more simple context. But we know that parasites often have very complex life cycles that move through multiple hosts and have very different transmission mechanisms happening. So before I get into my main research question addressing that, I want to introduce you to two parasites that have very distinct, very unique life cycle structures. So the first one is Halopagus. It is a fluke or a worm, and it uses 4 hosts to complete its life cycle. Hematoloecus is a very similar parasite. It uses similar hosts. It's also a fluke, but it has a 3-host life cycle structure. Now, both of these parasites infect dragonfly nymphs, pictured here, and that stage of their life cycle is going to be the one that I focus on for my talk today. And before I move on really quickly, I just want to mention that these parasites will be color-coded for the purposes of this presentation. Halopagus will always be blue or cool colors, and Hematoloecus will always be orange or warm colors. Now, the transmission modes of these parasites to their dragonfly host are very different. So Halopagus is exclusively a gastrointestinal parasite because dragonflies become infected with this parasite by consuming infected prey or zooplankton. Hematoloecus, in contrast, it's much more cosmopolitan. It can encyst or attach anywhere on or in the body of the dragonfly because Hematoloecus is acquired more passively from the water column, from free-swimming parasite larvae in the water. Now, these two different modes of transmission are the main difference that I want you to focus on here because that's going to be the main idea of my talk. Transmission mechanisms between these two parasites are very different, which is why I expect that cannibalism between their dragonfly hosts are going to differentially affect these two types of parasite. And that leads me to my research question. How does cannibalism between dragonfly hosts differentially affect the prevalence and the abundance of these parasites that have very different transmission mechanisms? And real quick, before I move on, I just want to say that dragonfly nymphs are a really great organism to use for this type of project. They are aquatic as nymphs, where they recruit their parasites, but they're also highly cannibalistic. And of course, they are host to both Halopagus and Hematoloecus parasites. So for my study, I set up mesocosms with these dragonfly nymphs. Dragonflies were collected from local ponds, placed in cups. 30 individual nymphs were counted out, and 30 nymphs each were placed into my green mesocosms pictured here. So I had 2 cannibalism treatments, 2 levels of cannibalism, low and high. which were accomplished by manipulating the size structure of those dragonfly nymphs that I put into the tanks. Low cannibalism tanks received dragonflies of all the same size to reduce predation, and high cannibalism treatments received size structured, so a mixture of small, medium, and large dragonflies. I then crossed those 2 cannibalism treatments with 3 parasite treatments, a Halopagus only, where I had Halopagus parasites in that tank, Hematoloechus only, and I had a treatment that had both parasites in combination. Then I replicated each one of these 6 treatments 6 times for a total of 36 mesocosm units. After 60 days of the experiment, all nymphs were removed from the tanks, counted out, and individually preserved for dissections. So all nymphs and any adult metamorphs that happened to emerge were dissected individually. Their intestines were extracted and dissected to look for Halopagus, while the whole body of the nymph was examined for Hematoloechus, because they do have different site selection in the host. Okay, and the very first result I got from this experiment was that the number of dragonfly nymphs remaining in each mesocosm had a lot to do with what cannibalism treatment they had. So you can see that for my low cannibalism treatment here on the left, there was much higher survivorship. So here on my y-axis, I have the number of nymphs left alive. There's a much higher number of dragonfly nymphs remaining in my low cannibalism treatments than my high. But the real story gets interesting when you look at the parasites inside of these dragonflies, right? Because that's a big part of my main question. So the first thing I'm going to show you for my results is parasite load per capita, or the number of parasites per dragonfly host. Now, for these figures, here on the y-axis, I have the number of parasites per host, per dragonfly, and on the x, I have my treatments. Hematoloechus is going to be on the left, and Halopagus here is on the right. For Hematoloechus, the take-home message of this figure here is that there really wasn't a huge difference between my treatments in terms of the number of parasites per dragonfly. So there was really no clear effect of cannibalism on Hematoloechus parasite burden. For Halopagus, the story was quite different. In my high cannibalism treatments here on the right in the blue, actually had more parasites per infected dragonfly. So higher cannibalism rates ended up leading to higher Halopagus parasite burden. But per capita parasite infection is not the entire story, right? I also wanted to look at the total number of parasites coming out of these communities. So the next thing I'm going to show you is total parasite abundance per tank, or the number of parasites in each community that I had set up. So again, here on my y-axis, this time the y-axis is the number of parasites per tank, per mesocosm, across all nymphs. And again, my x is treatment. Hematoloechus here on the left. The take-home message here is that here on the left, these are my low cannibalism treatments here. Low cannibalism led to a higher abundance total of Hematoloechus parasites, while for Halopagus, that relationship was somewhat flipped. Higher cannibalism here on the right in the blue led to a higher total abundance of Halopagus parasites in those communities. Now, the take-home message here is that cannibalism between hosts differentially affects these parasites based on their unique life history strategy and their different transmission mechanisms. So cannibalism decreased Hematoloechus success in terms of total abundance. There was little effect of cannibalism on parasite load per capita for this parasite, but it seems like the negative effects of cannibalism on Hematoloechus abundance was driven by the loss of hosts, because in my high cannibalism treatments, of course, I had much fewer nymphs survive and fewer hosts to carry that Hematoloechus parasite. In contrast, cannibalism was actually shown to be possibly beneficial for Halopagus at both the individual host and the community or tank level. So there was a higher infection load or per capita per host for Halopagus, and that could have been due to either these parasites are transmitting through cannibalism from dragonfly to dragonfly, or there was a reduction in competition for prey, because in my high cannibalism treatments, of course, there were fewer nymphs total left alive, thus there was more prey to go around per nymph. Also, there was higher parasite abundance in total at the community level for Halopagus, when I had my high cannibalism treatments in spite of host loss. So even though there were fewer hosts surviving in my high cannibalism tanks, I still had a higher number of parasites total across those nymphs, which was a really interesting and super cool result for me to get. So in conclusion, the life history strategies of these parasites and these pathogens that we share a world with are going to be really important when considering their ecology and transmission. Things like different transmission mechanisms are going to be really important to consider, and the effects of things like biodiversity loss and host population decline in our changing world are going to differentially affect these parasites based on their life cycle structures and life histories. So it's really important to take into account, especially for disease management strategies. So when we think about disease and parasite management in our communities, we really do need to think carefully about parasite ecology. And this idea has been implemented successfully worldwide with other parasites, which has been really successful across the world. And with that, I will go ahead and end my talk. I wanted to really quickly just acknowledge the people who have helped me out along the way, my lab mates, my advisor, Mike, and of course, my funding agencies, Sigma Xi, North Carolina Herp Society, IHS, and of course, NSF. I thank you so much for listening, and I welcome all of your questions. Thank you.